Good, yes. So I'm going to talk very quickly because I thought I had 35 minutes, I only got 20 minutes. And he's only given me 10 now because I moaned about not keeping other people to time. But I should be able to keep a narrative going, and I'm going to record this and put it on my YouTube channel, and they're going to record it and put it on their YouTube channel as well. So it actually goes very nicely with the previous talk, this talk, because I'm going to talk a little bit about ancient historical DNA. Obviously, there's a lot of promise here. We all know about Jurassic Park and all that stuff. And there were early successes in this field with thylacines and so forth, where, yeah, it was a thylacine. It was a historical sample. But there's this problem of reliance on amplification. You've got the risk of contamination. When you're making your amplification approach is very, very sensitive. You're going to pick up DNA from handlers. And, of course, with PCR, you've got amplicon carryover as a problem. But nonetheless, there have been some spectacular successes more recently with ancient human DNA. We've had whole genome sequences from a range of ancient human specimens. But we've also had spectacular failures. The first Neanderthal shotgun sequences, when they said, oh, they got a mega base of Neanderthal DNA, they subsequently had to say, oh, well, that 88% of that was modern human stuff that contaminated it. And if you're looking at things like Egyptian mummies, there seems to be this whole division among skeptics and believers. And I actually came across this quite forcefully over the summer. I was reading this book, The Shadow King, by Jo Marchant, who is a microbiologist who's now turned into a journalist and writes books on various things. She's written a book about Tutankhamun that was actually, her interest was piqued because she read an article in JAMA where they presented a load of analyses on Tutankhamun, and then she read a load of letters that followed where lots of other scientists completely trashed all the supposed findings in JAMA. And she points out here that basically there is a field split entirely in half. There's one camp, the mainstream, the biggest labs, argues that DNA from Egyptian mummies simply cannot survive well enough to be studied, and another camp's been publishing on this field, and they don't resolve the differences, they just go to different conferences and they publish in different journals. So it's just a weird, looking at it from outside, you just kind of think this is a slightly weird field. But there are lots of resonances with the previous talk. You know, all these people that believe there are brain microbiomes out there and bradyrhizobium enteric, and then there are the rest of us more rational individuals who believe it's rubbish. But I'm not going to rubbish the whole field of ancient and historical DNA and infection, because there is a substantial literature on all of these different diseases that have been detected in ancient and historical samples. And recently we have seen paleogenomics of the Black Death and of TB, using various capture-based approaches to get genomes out of there. There's less risk of contamination from DNA from the handler or the environment, because most of us haven't got tuberculosis when we're handling samples. We're not coughing our own TB onto the sample. But nonetheless, there is still this skeptic versus believer schism, even in this field, where you're looking at pathogen DNA rather than human DNA from ancient and historical samples. And here's a nice little ding-dong, which I actually reread this on the way here from a few years ago, where one group of people said, look, a lot of the stuff that's out there that's been published is nonsense. And not just nonsense, they were almost making the case that it was unethical to destroy archaeological samples to do such bad science, in their view. And then the group that responded here, including some of our collaborators, made the point that you're talking nonsense and we've done our experiments correctly, and there is this consilience of different kinds of evidence and so forth. So, if I'd have known about all this, I don't think we'd have got into this field, but I got approached by this guy here, Dave Milliken, and Helen Donoghue, who said that they had some old samples. David said he had some 17,000-year-old bison that he thought had TB in it. Could we get any genome out of it? I said, well, that's too precious for us to muck around with. Is there anything more recent that you've got more of that we can sort of play with and work out procedures? So Helen came in and said that she did have some material. Now, I said to them, look, actually, I'm not going to muck about with the PCRs. We don't have an ancient DNA lab. The only way we're going to do this is by shotgun metagenomics. And I was buoyed up by our experience with the JAMA paper that Nick described earlier, where we'd actually shown that metagenomics worked on human fecal samples. And there are obviously some reasons for doing metagenomics rather than amplification, because you don't have this problem of amplicon carryover. And there's much lower risk of even of unamplified pathogen DNA that may be around in the lab and in the environment. You don't have to do hybridization capture if you do real shotgun metagenomics. I mean, some people try and capture stuff and then call it metagenomics. But if you don't capture anything, you just sequence what's there, it's assumption-free. You'll find unknown unknowns. You'll find pathogens that you might not expect to be there. And also, it's less onerous in a way because you don't have to design and optimize all those capture codes. Now, sitting in your armchair planning the experiments, of course, there are still issues. The same issues that we had. Nick didn't actually mention this, but before we did those studies on the fecal samples from the German outbreak, we said, oh, well, we're just going to get 99% of it will be human DNA, or 99.9%, and the stuff we're interested in will be squeezed into a fraction of a percent. There is also problems of contamination with, I don't know if it was Nick or myself, when we used to kind of brainstorm at lunch and talk rubbish, we coined the term necrobiome. You know, wouldn't it be good to go to the body farm and actually sample the microbiome of patients, well, people, not patients, because they're not patients anymore, dead, aren't they? Dead people as they rotted away, and maybe there could be all sorts of, you know... What's that? Yeah, I guess so. I'm just trying to spread the nonsense. But anyway, we kind of thought, you know, you could get time of death and all that if you looked at the microbiome, or the necrobiome. And, of course, there's the problem of contamination of the soil microbiome. Here's a bit of philosophy of science. I just found this quote from Carl Sagan, which seems quite apposite here, really, that there is this tension in what we do between trying to be new and do exciting things, and then just trying to trash everything that's around us and say it's all nonsense. And it's these two processes together that, in his terms, winnow deep truths from deep nonsense. The other thing that we've been saying recently, though, is that because sequencing has become so straightforward and so easy, and with the benchtop sequences and so forth, actually, you shouldn't do too much thinking. Just get on and do the experiment. And here's a very old quote here from John Hunter that said the same kind of thing when he was talking to Edward Jenner. You know, why think? Why not try the experiment? And so I said, well, let's just extract some DNA from your samples and sequence it and see what we find. In fact, we were buoyed up. It was Helen that actually pointed out to me. I hadn't seen this paper. That actually there was one precedent, which was that when they sequenced this Tyrolean Iceman, sometimes called Ötzi, they sequenced his genome, but effectively they were sequencing a metagenome, and in that metagenome they found Borrelia burgdorferi, providing indication that this was the earliest human case of infection with the pathogen for Lyme borreliosis. And this was an interesting example of how this kind of approach could work. So Helen then introduced us to an interesting case study that we might look at. In this town of Vac in Hungary, there was a crypt opened up in the 1990s, and in there they discovered 242 bodies. For some reason, to do with the temperature and the atmospheric conditions, these bodies, instead of rotting away down to skeletons, had actually become naturally mummified. So there were large amounts of soft tissue left, even though they'd been in there for 200 years or more. And these included, for the purposes of the latter part of the talk, the remains of an individual called Teresia Hausmann, who died on Christmas Day in 1797, aged 28 years. So this is the church here where the crypt is found. Here are some of the coffins found in the crypt, and here are some of the mummified remains. These are not the individuals that I'm talking about now, but they just illustrate the way in which they're now being preserved and found. Now, some of these mummies had signs consistent with tuberculosis. And if you look at any of the kind of textbook descriptions of tuberculosis, they always tell you about that tuberculosis has been falling since records began. BCG had a little bit of an effect, and the proper antibiotics had a little bit of effect. If you extrapolate back, probably this time, around the end of the 18th century, was probably the time for peak tuberculosis, when it was at its worst in terms of prevalence. So various microbiological and molecular methods on chest sample from Teresia Hausmann confirmed a diagnosis of tuberculosis and actually provided some limited genotypic and quantitative data, basically suggesting that actually there was very good preservation of TB DNA in these samples. Been a number of papers published, largely with Mark Spiegelman working with others. This is Mark Spiegelman here, actually collecting samples from these mummies, taking care to do this aseptically, using an endoscope to go in there and take the samples. And one of the papers they published, they actually tried to grow tuberculosis from these samples, M. tuberculosis, but they failed. They tried doing this in broth culture. They tried even injecting it into guinea pigs, which is supposed to be the most sensitive method, although my career started with a letter saying that it wasn't, and that it wasn't worth doing. But anyway, they didn't grow it, but they did see acid-fast bacilli. Now, that's not absolutely certain to be M. tuberculosis, but certainly consistent with a high load, bacterial load of M. tuberculosis in these samples. And they've had another couple of papers published where they have homed in on these samples and actually detected M. tuberculosis DNA using amplification-based approaches, or PCR-based approaches. So we said, all right, well, we'll do the metagenomics then. So we did our first run on these samples up here. This is from a display in the museum in Hungary, where these bodies are kept, looking at what, trying to reconstruct what Terezia Hausmann would have looked like when she was actually placed in the crypt. So we studied this body 68, Terezia Hausmann, and Jackie Chan in my group extracted some DNA from lung tissue. She used a method which Helen Donahue had actually developed, in which you kind of marinate the sample for long periods of time in all sorts of noxious things and grind it up and whatever. It's quite a harsh method, which Helen told us was necessary to get DNA out of mycobacteria, because otherwise the cells are very hard to crack open. The sequencing library was made with Spry, works with this size selection, and we got 5.5 million reads using the 2 × 150 base pair kit. Of those, less than 1% aligned against the human genome. So our armchair idea that we'd have been swapped with human DNA was not true. And it does perhaps cast doubt on those who say you can't get DNA out of Egyptian mummies, because if you can't get it out of 200-year-old mummies, it's going to be even harder with Egyptian. Anyway, 8% of those reads aligned against M. tuberculosis. We just used the H37RV, the first genome that was sequenced from M. tuberculosis, as our reference. And we got an average 32-fold coverage of this genome, which is not bad for if you were doing this from a pure culture of the organism in a way. Now, I first of all said, well, this is just too good to be true. And scratched my head, racked my brain. How is this an artifact? Is there something gone wrong here? But we couldn't think of any reason why it could be. We hadn't done any culture of M. tuberculosis in our laboratory. We'd never performed any M. tuberculosis-specific PCRs. And the thing was, we achieved deep and even genomic coverage. So I can't think of any other explanation that this is real. It's not contamination. Having said that, there were other things in the sample. It wasn't all just M. tuberculosis. And Martin Sargent, who'd done some of the initial bioinformatics, he showed me these results here. But actually, M. tuberculosis was there, but also there was a nocardia and this thermo bifida. He actually did some assemblies and came out with various contigs. We don't think that the nocardia that was there was actually a known species of nocardia. It's not quite close enough to anything that's already been described. Nocardia does contain some human pathogens, the genus, and Martin pointed out that Bing Crosby actually died from nocardia infection. That's just as a slight factoid there. I don't think the nocardia is actually a pathogen here. I think it's a post-mortem contaminant. But it does open the question. I also looked in the literature, the number of cases of tuberculosis and nocardia in the same patient. I haven't been able to find any of those. So again, I think that's just a contaminant. This is the mapping data. You can see it's fairly even. There's occasional trough there. There's a big peak there, which is probably the 16S, which is finding all sorts of other 16S in the sample. But when Martin zoomed in, he actually found some indels in there when you compare against H37RV, and these indels were indicative of a particular lineage, this lineage called the Harlem lineage, which contains modern strains like the 7199 strain that came out of Germany recently, and another famous strain known as the Erdmann strain. Now, Martin then tried to draw some SNP-based phylogenies, and he couldn't get things to work, scratching his head and getting really quite stressed. And those of you who know Martin, his basal situation default is to be stressed. So here you could see his jaw muscles clenching and relaxing repeatedly until he came up with the answer, which was that he was using a SNP pipeline that filtered out SNPs that were present in less than 80% of the reads in an alignment. And when he took away that particular requirement, he suddenly realised that there were some SNPs in this metagenome, in this sample, that were present around 50-50. Quite a large number. So he concluded that he actually had a mixed infection here. There were two different genotypes of M. tuberculosis in this sample mixed together. And it just so happened that they appeared to be 50-50. I said, well, that's a bit of a, it seems a bit of a coincidence that they're exactly 50-50. But again, could we think of any reason, artifactual reason? We couldn't think of any reason why that should be. We just had to accept that that was the fact. Now, it turns out that the previous spoligotyping methods using these amplification-based approaches had also identified two genotypes in samples from this particular family, because in the crypt there's a mother and a sister of this individual as well. So that kind of gave some more credence to the fact that it was a mixed infection. And if you search literature, it turns out if you go to places like KwaZulu, where TB is quite common, about 9% of people actually have mixed infections. They have more than one strain genotype in their tuberculosis. And so from these analyses, we think it looks quite similar, actually, to this outbreak strain from Germany, from that particular lineage. Here's some, just again, you can see that in some parts of the genome, you've got SNPs that are sufficiently close together that you can see two SNPs on the same read. And you can see they segregate into these two different groups of SNPs. Again, evidence that there is this mixed infection there. In fact, when you look at coverage plots as well, there are some sections of the genome where you see these dropouts compared to H37RV, where that's deleted in both. But in other parts of the genome, you can see suddenly the coverage falls to about 50%, suggesting that these deletions are present in one of those strains in that mixture, but not the other one. So we've written this up, and it came out a few weeks ago as a letter in the New England Journal, and we tried to summarise our results here by showing that there are a number of deletions that happened after this lineage diverged from the lineage leading to H37RV, and the number of 300 SNPs, but then there were some additional deletions that happened between these two strains, and then there have been additional deletions in the lineage leading to 7199. Okay, well that was a one-off, and we got it to work. So we then went back and see if we could do it again. So Jackie took a different piece of lung tissue from Teretzia Hausmann. So it was the same individual, effectively the same sample, but it was just a different part of the sample. And she made the library in the same way. We got 19 million reads, less than 1% mapped to human again. This time we got 28% of them mapping to 7199, giving us a coverage of about 550-fold. Now, with that increased coverage, we can actually start to see that the SNPs are giving us ratios that are not actually 50-50. It looks like they're 45-55. And the process is underway, this is still work in progress, to try and bin them according to their ratios and reconstruct each strain genotype separately. And that work is ongoing. Now, I've got one more slide after this. Okay. Run 3, body 121, is a male who was a steward of an estate who died in a house fire. And again, we extracted DNA from this, and we did 14.85 million reads. Larger number of these actually map to the human genome. Now, we have got tuberculosis, well, we've got microbacterial sequences in there, but it's been very hard to sort of work out. It's only been going on for the last week or so, trying to work out how this maps to H37RV. It appears that there is an M. tuberculosis genome in there, and there is another closely related TB genome. I just did a simple, stupid experiment of just taking RPOB, a housekeeping gene, from M. tuberculosis and doing a blast search into it. And there are a number of reads there that have 100% identity and a large number that have 99. So I'm convinced we still have TB DNA in here, but it's going to take us a little while to sort this out. Stop press. Well, when we started this work, we thought we were going to be the first, but by the time we got around to publishing, we'd been scooped twice. There's a paper on Egyptian mummy metagenomics where they actually detected malaria and leishmania sequences, no, not leishmania, toxoplasma sequences in their metagenome. And then, of course, with much more fanfare, there's been this paper from Johannes Krausers and others where they actually managed to get a leprosy genome out of a tooth, from a tooth metagenome. We're going to do more of this kind of stuff. We have the badger poo or badger shit metagenomics project. We've got the TB genome out of badger shit. We're trying to do other ancient DNA work on human pathogens. We may or may not take that on. And we're doing some sedimentary ancient DNA stuff. That's me finished. Thank you.
Okay. I think we're going to skip questions.
Yeah.
All right. If anyone puts their hand up very quickly, you can have one question. No? No. Okay, I think we need to move on.
All right. There's no clock in here. Oh, there is a clock.
Yeah.